still has elevated intracellular inorganic carbon concentrations. That's what the gray bars are. The white bars are the amount of carbon that was fixed. So this plus this equals the inorganic carbon that's transported by the transporter, right? So the guy that has the mutation in the genes that are not related to the carbon concentrating mechanism acts like wild type, has a belly full of bicarbonate, and it fixes carbon quite merrily. The carboxysome interruption, we kept a couple of these around. It's able to transport bicarbonate, which you'd expect because it's the carboxysome that got ablated, not the bicarb transporter. It's able to acquire intracellular bicarb concentrations that are pretty high, but it can't fix it. That's what you'd expect from a carboxysome mutant. So our controls are looking great. Here are the site-directed. These two are, let's see. Yeah, these two are the site-directed ones. This is the random mutant. So the mutation in 853 and 854 both result in a lower concentration of intracellular inorganic carbon. And if it's not in your cytoplasm, you really can't fix it very well, right? So we were able to show that mutations in these genes had a pretty major impact on inorganic carbon accumulation, which is more evidence that we may have a bicarbonate transporter of two parts here, 853 and 854, those two proteins working together to transport inorganic carbon. So the next thing I did was take those two genes, 853, 854, and see what homologs were out there. Are there homologs to 853 and 854 found elsewhere amongst other bacteria? So I took a look, and they're all over the place. And they're usually right next to each other. So again, this is another piece of evidence that these two proteins work together. What's fun is that in a lot of these other microorganisms, they're found right next to genes that are involved in carbon dioxide metabolism. So the blue stuff, that's carboxysomes. So a lot of times they're near carboxysome operons in autotrophic organisms. What's really fun is that down here we have two different organisms that instead of using the Calvin cycle for carbon fixation, they use the reductive citric acid cycle for carbon fixation. The stuff in gray are oxidoreductases that use carbon dioxide as a substrate. And here we have 853, 854 homologs right next to them. How fun is that? So 853, 844 homologs are useful even if you're using a different pathway besides the Calvin cycle. Beyond the autotrophs, sometimes non-autotrophs become relevant, believe it or not. I also found homologs in a member of the Chlamydiae. There's a carbonic anhydrase gene right next to them. So again, more inorganic carbon metabolism. And then quite frequently in many organisms, you had these genes right next to genes encoding steps of purine biosynthesis. And if you remember your biochemistry, there are several steps in purine biosynthesis that require carbon dioxide. So these are about the acquisition of inorganic carbon. If you're a Calvin autotroph, if you're a reductive citric acid cycle heterotroph or autotroph, or if you are a heterotroph with a carbonic anhydrase or have to make your own purines. How cool is that? CO2 acquisition isn't necessarily about autotrophy.
And if you're an Archaea?
Yeah, there's some halophilic Archaeota that have it, and that's a big question mark to me. I'm like, what? And there wasn't anything in the gene context that would tell me what they're doing in those Archaea, but they have it. Okay, so instead of a family tree with just one phylum lit up, we're making some progress here, right? So I was really excited by these results. I said, okay, we're learning a lot about how autotrophic and other organisms interface with the inorganic carbon from their environment beyond just the cyanobacteria. This is cool stuff. So heartened by this, I said to JGI, I'm like, hey, you know, I found this really cool thing in Hydrogenia vibrio crudigenus that has implications beyond just itself. How about we take a look at some of the other relatives of Hydrogenia vibrio crudigenus to see what we might find there? So the JGI sequenced 12 more close relatives of Hydrogenia vibrio crudigenus. So these were formerly called Thiomicrospiras, but they've been taxonomically reassigned based on more information from supertrees that Rich Bowden and I constructed. They've been split into three groups, Hydrogenia vibrio, Thiomicrorhabdis, and Thiomicrospira. Long, hairy story there. Happy to describe it over beer. But anyways, we had all of these sequenced. Now these come from a variety of habitats. You've got more hydrothermal vent ones. You also have some that are found in marine sediments where there happen to be reduced sulfur compounds available to them. And some are from shallow vents as well. There's an extreme halophile in there too. So these are found all over the place. As long as you have oxygen, reduced sulfur compounds, and seawater, you're going to find organisms phylogenetically affiliated to these. So they're kind of relevant to things like the sulfur cycle and seawater. So we had,
also?
Huh?
Marshes also?
I would say so, yeah. If you sent me out there with a plate of Thiomicrospira medium, I could find you some Thiomicrospiras. Oh yeah. Yeah, they're amazing. They're petri weeds. They grow really quickly.
Do they need salt, or do you find them in freshwater, like aquifers, too?
There was a report of a freshwater Thiomicrospira relative, so you can find them, but they seem to be much more abundant in marine systems. But when Jim Geary takes a look at some of his sulfide-containing waters from his caves, he'll come up with some Thiomicrospira hits. So they're there. So I had some more of these guys sequenced because I was curious about inorganic carbon metabolism and other things as well. I was expecting the story to be reasonably straightforward. I was expecting to find more homologs of 853 and 854. But anytime you make a prediction about nature, it kind of goes, ha ha ha. Yeah. It has its own agenda, you know what I mean? So some of these organisms, here's Hydrogenia vibrio cunegenus and its two-component, what I think, transporter, more evidence on that later. Some of these guys have that. This is their carboxysome operon. I'll start off by saying the carboxysome operon for these organisms, the synteny is pretty conserved amongst all of them. So the blue stuff is all the bicarbonate shell proteins. They're color-coded by the shell protein that they are. Here's the carbonic anhydrase. Notice this down here. So remember how I mentioned that with the carboxysomes, you've got rubisco in there? But then you've got the carbonic anhydrase that helps the rubisco use the high intracellular bicarb pool because the carbonic anhydrase helps hopefully convert some of it to CO2. Some of these guys don't have the carboxysomal carbonic anhydrase. They've got this thing, and I call it this thing because if you take this gene and at a nucleic acid or amino acid level, or if you try to read all 6 frames of it, if you try to compare it to all the genome and genetic data we have out there, there are no homologs outside of these organisms. So they don't have the typical carbonic anhydrase. They don't have something that's homologous to anything that's been characterized yet. This might be some sort of funky new carbonic anhydrase, or it could be something completely different. So as much as we think we understand carboxysomes, we might not. Cue the scary music. There's kind of a question mark right here. So there's what you see from genome data, and then there's what you think might actually happen in a living cell. Whenever you find something strange, it's good to confirm the truth of it by looking at the actual cells. Now the list here looks pretty complete, but this isn't everybody that I had sequenced. There was Thiomicrorhabdis arctica, as well as Thiomicrorhabdis, which one was it? Melos T2, and they did not have carboxysome operons. I'm like, oh, so not all of these guys have a carbon concentrating mechanism. So I took those 2 species and I tried to grow them under low CO2 conditions, and they didn't want to grow. But I wanted to verify that they lack carboxysomes, and everybody else had them, especially these. I wanted to see if these guys had carboxysomes. So I did the requisite electron microscopy, and everybody that had a carboxysome operon encoded in his genome, if you cultivate them under low CO2 conditions, they get freckles. The freckles are carboxysomes. Now here's Phyla, 2 freckles down here. That's the one that's got the funky carboxysome operon that's lacking the carbonic anhydrase gene. It still makes carboxysomes, and it's still able to grow okay under low CO2 conditions. So there's something weird going on with that one and its friends that are phylogenetically affiliated. The 2 that did not appear to have carboxysome operons in their genome sequence, if you look at them under EM, what it did was, since they did not want to grow under low CO2 conditions, I just exposed them to low CO2 conditions overnight. They're still acidifying their medium, but there's no increase in biomass, so they're just marking time. There's no carboxysomes. So I was able to confirm the genome data with electron microscopy. So we've got a little bit of diversity on the carboxysome front, but the really weird diversity, the one that really shocked me, was the stuff beyond the carboxysome. So here's Hydrogenia vibrio cunegenus. These are the 2 genes encoding the 2-component bicarb transporter in that organism. Now notice that the T, I'll just clarify, the T means transporter or predicted transporter. Notice everybody else has T's after their carboxysome operons too, but they're different colors. That's because they are 4 evolutionarily independent transporters. So transporters are described in a large array of families. Each of them are evolutionarily related to each other within a family. So we have 4 different evolutionarily independent families of transporters represented downstream from the carboxysome operon. That actually surprised me because these organisms are all pretty closely phylogenetically related to each other. I wasn't expecting to see this much difference among them, you know? So, okay, we've got 4 different evolutionarily independent transporters downstream of their carboxysomes. Let's take a look at those different ones. I had Tara Harmer work with me to get some decent phylogenetic analyses done on these. So here's the BIC-A. Now the BIC-A transporter is found downstream, in, let me see here, KP2, that's this one here. It's also found in Hydrogenia vibrio and such. Now, the BIC-A is interesting because that's one of the transporters that you find in cyanobacteria. Not present in Hydrogenia vibrio crucigenus, but it's present in these guys. Now, when I saw that, oh, it's got a BIC-A. Maybe there's an HGT here. There's a horizontal gene transfer from the cyanobacteria. No, it's not, because the top 3, all the way up there on that hugely long branch, this is a maximum likelihood tree, so the branch lengths are a function of genetic distance. So the top 3 are the biochemically characterized bicarbonate transporters from cyanobacteria. Lots and lots of distance. Here's the ones from the organisms that I had sequenced. They're very distantly related to them. Now the thing about transporter genes is you can't really tell what the substrate is for the transporter based on its membership in a family. So even though something's in the same family as a biochemically characterized bicarbonate transporter, within a particular family, you're going to have transporters that transport all kinds of things. Given the distance between the bicarbonate transporters that have been characterized and the ones that I found, I couldn't really make assumptions here. So distant homologue of a cyanobacterial bicarb transporter. One of the other transporter families was the SBT family. Again, this is a bicarb transporter you find in cyanobacteria, but just like the previous, it's too phylogenetically distant from the cyanobacterial version to make any sort of, I don't know, prediction that you feel good about that they transport the same thing. So very distantly related. The third kind of transporter I found downstream from the carboxysome operon was the chromate family transporter. Now chromate, as you can tell from the name, chromate family transporter, has been described as a chromate transporter in the organisms for which this is biochemically characterized. Here's the Thiomicrospirus. All the way over here are the ones that are biochemically characterized. So it belongs to the same family, but again, I can't really say, oh, it's a chromate transporter because it's too distantly related for me to feel good about that prediction. Okay, so I've got these 4 transporter families that are encoded downstream from carboxysomes. I wanted to collect more information to convince myself whether or not these actually transport bicarbonate or not. One line of evidence was to do some qPCR and compare the transcript abundances from these genes under low CO2 versus high CO2 conditions. You'd anticipate if it's part of a carbon concentrating mechanism that these genes are going to be transcribed, or the transcript levels are going to be way higher when the cells are cultivated under low CO2 conditions. So I grew everybody in chemostats under low and high conditions, isolated their RNA, and did the appropriate qPCR, and I found that all these transporters, by and large, are lit up, the transcript abundance is higher under low DIC conditions. So, okay, another line of evidence that perhaps these might transport bicarbonate. We're getting there. So I wanted to actually put together an ironclad story that these guys are taking up bicarbonate, and all of the stuff that I presented so far is mostly guilt by association. So what I did was I picked a member of each transporter family, and I preferentially selected them from organisms that we can manipulate genetically for downstream studies. So I grabbed the SBT transporter from KP2, SULP, and the chromate transporter, in quotes, from JR2, and then the 2-component transporter I previously characterized to the best of my abilities from Hydrogenia vibrio crucigenus. And took these, I amplified them out, and I put them into an expression system. So this was my what I did last summer project, and I didn't want to give it to a graduate student because expressing transporters and E. coli heterologously is a nightmare. The internet is painted with horror stories of semi-suicidal graduate students and postdocs who've tried to express transporter genes in E. coli. And the reason why it's such a headache is that anytime you move a gene, you've got a choice about what promoter to put it after. A common one to put it after is a T7 promoter, but the T7 promoter is a really strong promoter. You put a gene behind a T7 promoter, and it's just going to be raining RNA. It's just, ugh! It's a really strong promoter. Now, the problem with that is that if you make too much of a particular protein, especially something really hydrophobic like a transmembrane protein, it's all going to form insoluble aggregates inside the cell and kill the cell. That's kind of a bummer if you want to do physiology. Dead cells are not good physiological subjects. The other problem is, even if it doesn't kill the cell, cells have a special system they use to make sure that integral membrane proteins get inserted into the membrane. It's called the CQIE system. So if you overexpress an integral membrane protein, the CQIE system is going to be so busy with your protein that it's going to forget about other important stuff like ATP synthase. So you're going to kill your E. coli that way. So it's really hard to express transmembrane proteins. So I found a system where, yeah, it's behind a T7 promoter, but you can attenuate the activity of the T7 promoter by including a gene encoding an inhibitor for the T7 RNA polymerase. So you can dial it back a little bit. So you're not going to form aggregates, and you're not going to saturate the CQIE system, and your E. coli have a chance of surviving the expression of these weird genes. So I moved the genes from my thiomicrospira relatives into E. coli, expressed them under a system where you could dial it back a little bit, and then I did siliconyl centrifugation to see if I turned E. coli now into cells that could accumulate intracellular inorganic carbon. E. coli can't do that. So I figured a gain-of-function assay would be a really great way to prove once and for all that these are actually bicarb transporters. I've just started running these experiments, and I got really, really freakishly lucky. I told Maya I'm going to stop doing science now.
Well, you're ahead.
That's the last time anything's going to ever work for me. I've got this bad feeling in my bones about this. So this is the construct that had the 2 subunit component in it from Hydrogenia Vibrio prunigenus, the first one I described. F means it's in a forward orientation, so it's in the correct orientation to be expressed from a T7 promoter. R, I got lucky when I was making these mutants. Some of them wound up backwards. I thought, hey, this will be a cool negative control. So these are in reverse. So if you compare the intracellular inorganic carbon for the forward orientation versus the reverse, it's boosted here. I'm going to be tinkering with this to see if I can make it talk a little louder, but this is still already pretty convincing. SPTF is just ridiculous. It's off the chart here. SULP, the BIC-A, this one also. The chromate transporter, it hasn't woken up at this point as an inorganic carbon transporter, but I'm not going to give up on it yet. There's a few more things I can tweak it with to see if I can get it to wake up as a bicarb transporter. But still, 3 out of 4 ain't bad. I wasn't expecting even one of these to work. It was just kind of this thing I was going to do over the summer to keep myself out of trouble. So this actually worked, much to my shock. Like I said, I'm going to set down my pipetters now. So now, instead of just one phylum of autotrophs, where we understand how it interfaces with the inorganic carbon pool in its environment, which is kind of an important part of the ecophysiology of autotrophic organisms, in my opinion, instead of just one phylum, we now have a little bit of understanding about 17 phyla of autotrophs. How cool is that? And then this funky thing from the Archaea. So we've gone from this to this, and that's pretty cool. So hopefully we can have more insight into how these organisms function in the really crazy array of habitats where they form a foundation of primary productivity, but also catalyze reactions of geochemical importance. So why is this exciting? I think it's great because it's a really neat evolutionary story. We have these rather closely related organisms that all have very divergent ways of taking up inorganic carbon from their environment. It's going to be fun to do some really hardcore biochemistry to characterize these different transporters to see what their differences are, because they're not going to be doing the same thing the same way. They're going to have different affinities. If they're a symporter, they're going to have different stoichiometries that'll dictate the size of the intracellular pool of inorganic carbon that can generate. There's all sorts of cool follow-up questions here. So there's the evolution part of things, which makes me excited as a biologist. And then for those who are a little more practically minded, there's some biotechnological applications too. We're trying to figure out ways of tinkering with microorganisms to have their biochemistries produce target compounds of industrial interest, and there's a lot of interest in making carbon-neutral biochemistries. A lot of people are trying to take autotrophic organisms and make them make compounds of industrial importance. Well, if you front-end that process with a bicarb transporter, you're going to probably make a more robust system that's going to be able to do the biochemistry you want it to, even if the pH goes all over the place, and even if the inorganic carbon concentration around it is not the stablest thing in the world. So there might be some cool applications to this as well. Okay, that's my story. So gratitude. I'd really like to express gratitude to the army of graduate and undergraduate students that I've had working on this project and providing their insights and their patience, especially in the making of that giant library. Thank you for listening. Maya.
Okay, so in your